Okay, rare event in paper setting. We've got another question on spermatogenesis. So, looking at specifically, it says it's controlled by a number of hormones, so that's what we're going to be looking at. Attempts to develop a male contraceptive pill have focused on disrupting the sequence of processes involved in so we want to mess about with male hormones so that they're presumably not making sperm. That's the only way it would work, isn't it? So the photomicrograph shows a section through the seminiferous tubule of a mammal. So here we've got the outside of the tubule, and here we've got the lumen. And you can see we've got dividing cells here, various stages, and we've got another one there, so this is the bit in between our tubules. So, this is the gap, so I'm thinking these are Leydig cells. These are, they've got tails, so they're spermatozoa. We've got the germinal epithelium around the outside. Spermatogonia, you'll sometimes see those cells called. And then here we've got quite a big chunky cell. That's probably a primary spermatocyte maybe. It's quite near the outside. It's clearly it's in this layer of cells that are undergoing cell division. And then we've got lots of these little small black dots. I think they're going to be spermatids. I wonder if that one might be a secondary spermatocyte, because I've got one that's dividing over here. It's in prophase, look, and it seems to be a bit closer to the outside. So remember that those sperm gets pushed inwards, don't they? So let's say that that one's a primary spermatocyte, and then we've covered all bases. And sometimes, you can't recognise these cells just by looking. You have to look at their relative positions. So, use the letters on the diagram to identify the following. A cell that secretes testosterone. So testosterone I know is from the Leydig cells. That's O. A gamete, and our spermatozoa. A cell that will divide by mitosis. P, germinal epithelium. The cell that must differentiate to become fully functional. So this is our inside one that's going to be a little blob of cell differentiating into a sperm. K. That was a good one. So, the flow chart shows how sperm production is controlled by hormones. FSH, so we've heard of that, that's the female one. Stimulates Sertoli cells to initiate spermatogenesis. At this point I think it's worth remembering the context that we're trying to prevent sperm production. That's what that very first bit said, it's working on the male contraceptive pill. Luteinizing hormone, LH, stimulates the production of testosterone, which is involved in controlling the formation of spermatids. So there we've got a sort of two-pronged approach, haven't we? We could stop FSH stimulating Sertoli cells, and therefore we wouldn't have spermatogenesis, or we could stop LH making progesterone, which would mean that the formation of spermatids wouldn't happen. So here we've got that handily in a diagram for us. Pituitary gland, FSH acts on primary spermatocytes and Sertoli cells, presumably. So Sertoli cells, and then they're initiating that stage of development. And testosterone, controlled by LH, which finishes all that process off. Metronidazole is an antibacterial drug that's been shown to have negative effects on spermatogenesis, which is what we're trying to accomplish here. Study on rats. And the effect of the drug on the hormone levels gave the following results. So we're going up in dose, FSH levels are going down. That's the overall trend. LH levels going down. Testosterone levels are going down. Okay. Nice little gap for a calculation. What was the mean percentage decrease in FSH plasma? So we're only looking at FSH. And we're only looking at 200. So we want to know what's the difference between no drug and 200 milligrams as a percentage. So the difference is 12.07, take away 7.81. We want to know what it's decreased from, 12.07, and we need to make it a percentage. So 12.07 take away 7.81 equals, shared by 12.07 equals times 100. I've got 35.294. So I'm thinking this is to 2 decimal places. I need my answer to 2 decimal places. 29. Oh, another calculation. Calculate. Calculate the mass of a drug that would have to be administered to a rat with a mass of 550 grams to achieve a relative dose of 400 milligrams per kilogram. Okay, so 550 over 1,000 for the kilograms, multiplied by 200 to get it to milligrams, times 200. I don't know why I'm not now timesing it by 2. $550 over 1,000. Oh, because of the 1,000 thing going on. 550 divided by, I was just thinking it's about half a kilogram, but never mind. Times 200 equals 110 milligrams. And that makes sense, doesn't it? Because actually this is it is half a kilogram, and so we need about half the dosage of the drug to make it up. So doing a quick mental check is always a good idea. Okay. The scientists concluded that the drug was acting on a part of the brain. Using the information given to explain why they came to that conclusion. So what do we know? FSH and LH. What's this got to do with the brain? FSH and LH are pituitary hormones. The pituitary is part of the brain. So I know that sounds really obvious, but you've still got to state it. So how does it act then? So FSH decrease. Decreases, so there is less effect on using this diagram, because it says use the information, so we've got to use that and that. Less effect on the primary spermatocytes. So less meiosis. So they don't do meiosis 1. And then we've got the LH1. So the LH decreases. Which would result in less testosterone. And therefore less secondary spermatocytes undergoing meiosis. Or you could have said, producing spermatids. And I think you could probably also perhaps talk about less FSH, so less initiation of spermatogenesis. Less FSH means fewer Sertoli cells, because you told that as well. Initiate spermatogenesis. Okay. Based on the evidence given, suggest 2 other sites where metronidazole may have been acting, giving reasons. So, 2 other things that we've mentioned. So remember, this is scaffolded. You can go back to the beginning of the question and see what cells have we not mentioned yet. So we've talked about primary spermatocytes, secondary spermatocytes, spermatids. We haven't talked about Leydig cells. We haven't talked about Sertoli cells, apart from there where we say they're not initiating it. So instead of acting on FSH, they could have been acting on the Leydig cells. So could act on Leydig cells directly to prevent testosterone release. Or on Sertoli cells to prevent sperm being nourished during spermatogenesis. Okay, very good. Onwards and upwards.